So the topic for this video, for revision for this video is the mode of action of antibiotics or the chemotherapeutic agents. So these are the major way in which the antibiotics attack the microorganism. So first one is the inhibition of cell wall synthesis. Second is they damage the cytoplasmic membrane of microorganisms. In third way, they inhibit the synthesis of nucleic acid and proteins. And finally, they inhibit some specific enzyme systems. So firstly, talking about the inhibition of cell wall synthesis. In this case, the antibiotics, they inhibit the biosynthesis of the peptidoglycan cell wall structure. The examples of the antibiotics that function through the inhibition of cell wall synthesis are penicillin, cephalosporin, cycloserine, vancomycin, and bacitracin. Firstly, talking about penicillin. Penicillin are colorless in pure crystalline form. They are produced by the microbes Penicillium notatum and Penicillium chrysogenum. So these penicillins we all know were first isolated by Alexander Fleming in 1929. Their selective action is towards gram-positive bacteria, spirochetes, and gram-negative diplococci, which is Neisseria. These are a class of beta-lactam antibiotics. The beta-lactam antibiotics are the most important class of antibiotics in for clinical use. And the penicillins, all the penicillins, they have a base nucleus, which is the common structure. They have a common base nucleus and they have a fused beta-lactam thiazolidine ring in which there are different side chains attached to it. The natural penicillins, they are prepared through the salts of sodium and potassium. Uh, these natural penicillins, they are activated by heat, cysteine, sodium hydroxide, and penicillinase enzyme and HCl. These are not affected by the action of saliva or bile. Penicillin 5 is the antibiotic that shows greater stability than the other kinds of penicillin in the presence of acids. The common base nucleus in the structure of all kinds of penicillin, whether it is natural or the other kinds, is 6-amino penicillinic acid. And this 6-amino penicillinic acid is produced by interrupted biosynthesis in P. chrysogenum under the conditions that are required. And this is the process. The interrupted biosynthesis by P. chrysogenum is the process which is used for the production of semi-synthetic penicillins. P. chrysogenum gives penicillin G, which has a base nucleus of this 6-amino penicillin acid and side chain. The side chain can be removed by penicillin G through the use of amidase enzyme. The first ever semi-synthetic penicillin for clinical use that was created were the phenethicillin, and they are easily absorbed in the system as compared to penicillin 5, and they are equally effective as penicillin G. Methicillin is an example of a semi-synthetic penicillin, and it is more resistant to the enzyme penicillinase, and it is less likely to be inactivated by the enzyme, the methicillin. Talking about ampicillin, ampicillin is also a type of semi-synthetic penicillin, and it acts against a broad spectrum of bacteria. It is bactericidal, kills the bacteria. It lacks any toxicity to the host organism that is using it, and it's not resistant to penicillinase, and it is stable in gastric acid. That is, we can administer it orally and it will pass through the GI tract following its action. The mode of action of penicillins is as follows. The penicillins, they interfere with the final stages of the synthesis of peptidoglycan layer, and in this case, a transpeptidase reaction that happens for the during the synthesis is inhibited because the cross-linking of polymers of this peptidoglycan does not happen, and due to which the cross-linking of two polymer layers does not occur. So due to this the cell wall synthesis is inhibited. And penicillin are bactericidal to all the growing cells as they do not let the formation of peptidoglycan layer. So this is the structure of the base nucleus of the penicillin core, which is the 6-amino-penicillinic acid. This is the entire structure, and here is the side chain attached to it. The next kind are the cephalosporins. Cephalosporin also act through the inhibition of cell wall synthesis. They are produced by marine fungus, which is Cephalosporium acrimonium, and these are bactericidal to all the growing cells. The therapeutic uses their action against gram-positive and gram-negative bacteria, and they have a antibacterial property that is similar to the semi-synthetic penicillin. They possess low toxicity, and their action is they inhibit the cross-linking of transpeptidases, similar to the penicillins. Cycloserine is the next kind. It is the structure of cycloserine is related to that of alanine. As we can see, D-alanine structure is here, and the cycloserine is the cyclic structure. The cycloserine is manufactured through chemical synthesis, and it was discovered by as production by Streptomyces species, and mainly cycloserine is used for the treatment of tuberculosis. It is used in tuberculosis therapy, and it has significant side effects. That's why the use of cycloserine is limited. The mode of action of cycloserine is it inhibits or interferes with the synthesis of peptide moiety of the peptidoglycan, and both the enzymes that are involved in the pentapeptide side chain formation, the enzyme named alanine racemase and D-alanyl D-alanine synthetase, these both enzymes are inhibited by cycloserine. So due to this, the pentapeptide moiety of peptidoglycan is not formed, it is interfered, and the cell wall formation is inhibited. Bacitracin is the next kind of antibiotic. It is a polypeptide formed by Bacillus subtilis, and it is toxic to animals and human cells, so we cannot use it for systemic chemotherapy. And it's the only application of bacitracin can be in the topical treatment of infections that are caused by gram-positive bacteria. The mode of action of so orally we cannot administer bacitracin because it's toxic to us. The mode of action of bacitracin is that it interferes with the regeneration of monophosphate form of the bactoprenol from the pyrophosphate form, which is also known as the lipid PP form. Next is the vancomycin, which we obtain from Streptomyces orientalis, and it consists of amino acids and sugars. This vancomycin inhibits the peptidoglycan synthesis and binds through the binding of D-alanine D-alanine group on the peptide side chain of the membrane bound intermediate. So because of this binding of vancomycin to this group, this inhibits the peptidoglycan synthesis. So this is it for the video. I hope it helped. Thank you so much.